In this experiment, we extracted DNA from a banana. DNA extraction is important since this process allows for the study of structure of DNA.
First, create a salt, soap, and water solution. Add it to the bag and smash it with a half of a banana. Crushing the banana exposes the cells to the solution and physically breaks down the cell wall of plant cells.
Here are the materials you will need to complete this experiment. The dish soap will break down the cell membrane and release the contents of the cell, exposing DNA. Soap consists of a polar head and a nonpolar tail. The nonpolar tail of the salt will bond with the nonpolar tails of the lipids in the plasma membrane, forming a micelle. This causes the plasma membrane to dissolve in water. The polar heads are in contact with the water, and the nonpolar tails are bonded on the inside of the micelle. Next, you will add 5 milliliters of cold isopropyl alcohol.
The salt aids in clumping the DNA together. The sugar phosphate background of the DNA consists of a negative phosphate group and a deoxyribose group. Salt neutralizes the charge of the phosphate group, allowing the molecules to bond together and clump. The next step is to strain the solution through a sieve into a separate container. This removes any large chunks of fruit, leaving only the cellular components of the cell, water, and soap solution behind.
Temperature of isopropyl alcohol will be cold is important to separate and clumping the DNA. The cold alcohol allows the enzymes that break down DNA to be slowed. Alcohol is made up of nonpolar molecules. Hydrogen bonds form between water and alcohol, allowing sodium chloride to bond to the DNA and cause precipitate. Alcohol is needed because DNA is insoluble in the alcohol, and the alcohol is less dense than the aqueous solution. The alcohol will rise to the top of the solution, carrying up the precipitated DNA.
We were both successful as we carefully went through the DNA extraction. As an aspect that was important that we both did was mashed up enough fruit to properly see the DNA come to the top of the solution.
One aspect that can be improved is to slowly strain the solution into the cup to ensure the solution does not spill over the sides.